Hello everyone, I'm Higon Kim from Renatus. I'm very excited about having the opportunity to tell you about our approach to cholesterol-targeted therapies. Dysregulated cholesterol homeostasis and excess cholesterol accumulation are commonly observed in many age-related and metabolic diseases, including obesity, chronic kidney disease, neurodegenerative diseases, MASH, and atherosclerosis. The accumulation of cholesterol in various cellular compartments, particularly in lysosomes, plays a crucial role in the pathophysiology of many diseases. As a central hub for cholesterol trafficking, lysosomes are essential for maintaining cellular cholesterol homeostasis. Notably, cholesterol buildup in lysosomes disrupts key pathways, including mTORC1 activation, which leads to autophagy dysfunction, mitochondrial dysfunction, RS generation, lipids accumulation, and inflammation. There are some drugs available for cholesterol intervention, such as statins, ezetimibe, PCSK9 inhibitors, and fibrates. However, these drugs primarily target circulating LDL cholesterol levels rather than directly targeting intracellular cholesterol in diseased tissues. Cyclodextrin is a ring-structured molecule that can physically encapsulate cholesterol and solubilize it, allowing direct targeting of cholesterol. Cyclodextrin acts as a cholesterol modulator by forming complexes with excess intracellular cholesterol and promoting its efflux through reverse cholesterol transport and passive diffusion. One of its key downstream effects is mTORC1 inhibition, leading to normalization of lipid homeostasis, mitochondrial function, and inflammation. Let's explore why directly targeting and reversing cholesterol accumulation is crucial in disease contexts. Diabetic kidney disease is a complex condition driven by multiple factors involving metabolic dysfunction, hemodynamic changes, and inflammation. As shown in the images on the right, cholesterol dysregulation and accumulation are prominent features of diabetic kidney disease. Although they may be the underlying cause of the disease, these abnormal cholesterol conditions are currently not druggable. Importantly, statins, which lower circulating LDL levels, do not improve the prognosis of diabetic kidney disease. This is a mouse model with type 2 diabetes, which develops diabetic nephropathy. Compared to normal mice, diabetic mice showed significantly elevated cholesterol and triglyceride levels in the kidney tissue. Treatment with RNO5, one of our drug candidates, normalized the cholesterol levels in the diabetic mice. It also had a significant impact on lowering the triglyceride levels. Albuminuria was decreased by about 40% and blood creatinine by about 30%. These results show that cholesterol is a very crucial target in treating diabetic kidney disease and underscore the need for developing novel cholesterol-targeted therapies. Cholesterol modulation has shown promising results in other cholesterol-related diseases. In an Alzheimer's disease model, our cholesterol modulator facilitated the clearance of amyloid beta and significantly improved behavior in both zebrafish and mice. In metabolic dysfunction-associated steatohepatitis, or MASH, our cholesterol modulator effectively lowered liver fat levels, which was comparable to resmetirom, the first FDA-approved drug for MASH. In atherosclerosis, a cholesterol modulator effectively inhibited plaque progression and reduced macrophage content in the plaque. Our pipeline includes a broad range of disease areas, including chronic kidney disease, Alzheimer's disease, Niemann-Pick type C disease, atherosclerosis, MASH, and cancer. Hydroxypropyl beta-cyclodextrin is the first-generation cyclodextrin to be used as a drug and is currently in clinical development for Niemann-Pick type C, Alzheimer's disease, and atherosclerosis. Its mechanism of action, as mentioned before, involves targeting intracellular cholesterol. However, it is known to target cell membrane cholesterol as well, which can disrupt the membrane structure and cause toxicity. Also, hydroxypropyl beta-cyclodextrin has a higher affinity for other lipids, such as fatty acids, than cholesterol. These factors show the limitations of the first-generation cyclodextrin. Renatus cyclodextrins, on the other hand, allows more effective targeting of intracellular cholesterol. Unlike hydroxypropyl beta-cyclodextrin, Renatus cyclodextrins do not target cell membrane cholesterol and exhibit higher selectivity for cholesterol over other lipids. Excess cholesterol is present in cells as cholesterol crystals. You can see Renatus cyclodextrins exhibit superior ability to solubilize cholesterol crystals compared to hydroxypropyl beta-cyclodextrin. However, hydroxypropyl beta-cyclodextrin has the highest tendency to extract cholesterol in the cell membrane and induce hemolysis due to membrane disruption. Renatus cyclodextrins, on the other hand, are designed to avoid targeting cell membrane cholesterol, enabling the selective removal of harmful intracellular cholesterol while reducing adverse events. Although cyclodextrins are used as cholesterol-targeted therapies, they can bind to various hydrophobic molecules besides cholesterol. The lipid solubilization profiles demonstrate the selectivity of cyclodextrins to different lipids. Hydroxypropyl beta-cyclodextrin has the lowest selectivity for cholesterol, as it exhibits the lowest cholesterol solubilization while solubilizing other lipids more efficiently. Interestingly, although Renatus cyclodextrins exhibit better selectivity for cholesterol, differences in lipid binding exist among them. These variations suggest that each Renatus cyclodextrin may have a differential mode of action in the context of cholesterol modulation, making them valuable candidates for testing in different cholesterol-related disease conditions to optimize therapeutic potential. The physicochemical properties of Renatus cyclodextrin improve not only therapeutic efficacy but also the safety of the treatment. Ototoxicity is the dose-limiting toxicity of hydroxypropyl beta-cyclodextrin. It elevates the hearing threshold and causes outer hair cell loss in the cochlea, which is not reversible even after stopping the treatment. Iron 5 treatment at the same dose does not induce such an effect. Even at an approximate lethal dose of hydroxypropyl beta-cyclodextrin, Iron 5 causes no mortality, showing its superior safety. To summarize, compared to the conventional cyclodextrin, Renatus cyclodextrins exhibit superior cholesterol solubilization, avoid targeting cell membrane cholesterol, and has higher selectivity for cholesterol. We are also exploring ways to conjugate targeting agents to cyclodextrins for disease-specific targeting, such as in the brain. Overall, our next-generation cyclodextrin represents a significant advancement in cholesterol-targeted therapy, offering enhanced efficacy and safety. Renatus aims to make a critical impact on cholesterol modulation, paving the way for safer and more effective treatment options for patients. If you have any questions or suggestions, please contact us. Thank you for listening.